Hello friends, welcome to today's session on separation techniques. So in this session, we will be discussing about the basic techniques of separation. Along with that, we will be discussing about centrifugation. Okay, so before going to discuss about these separation techniques, I would like to give just a brief background of this. So since we are dealing with biochemistry, so here we need to do lot of assays and estimations and analysis. So before going to do how we are going to do all these assays estimations, let us know something about the basic techniques which are involved in separation of biomolecules as well as your subcellular organelles. Yeah, let us start. Before going to discuss the techniques, I would like to give a brief introduction about a mixture. So if you look at this composition here, you can see this structure which is a mixture. So mixture of few substances you can say. So any mixture which is made up of more than two substances, we call it as a mixture. To separate such mixtures, we are going to use techniques which are called as separation techniques, and these mixtures generally are of two types. So one is homogeneous, another one is heterogeneous. Homogeneous means the one which is made up of similar kind of substances. Heterogeneous means the one which is made up of different kinds of substances. In biological systems, we have such mixtures which are either made up of homogeneous compounds or made up of different substances. Okay, and moving further, so if you see here, there are various types of separation techniques which are like very basic techniques of your crystallization, decantation, evaporation, filtration, and fractional distillation, magnetic separation, as well as precipitation, and then sieving. Among these, if you see the red ones, which are like centrifugation, chromatography, and electrophoresis, so these are the techniques which are having high importance in the view of biochemistry. But however, the other techniques which are listed here, like crystallization, decantation, precipitation, and your sieving, these are the very basic techniques which we use frequently in our domestic life. So if you see here the sieving technique which we generally use at our homes to separate the or to clean our floor. At the same time, the other one is if you see the filtration. So in filtration also, what we do just when you are preparing some decoctions like coffee, tea, whatever it may be, so you use normal filters to filter solids from liquids. Okay, and similarly, if you see the decantation also, so in decantation also, without disturbing that precipitate, we are going to pour the what is called as top layers of the supernatant. That is called decantation. The other one is evaporation. So in evaporation also, what we are going to do, so we are going to boil the mixture. So generally in this evaporation, the mixture is composed of both solid and as well as the liquids. So when you boil this mixture, what will happen? The liquid substance is going to be evaporate and solid will remain in the dish. And similarly, if you see the other one, which is here magnetic separation. So magnetic separation, as it clearly says, so we are going to use the magnet to separate a mixture which is having non-magnetic material as well as the magnetic material. For example, generally when you observe during your childhood, when you are playing with a magnet, you can easily attract the metals as well as these what is called as rust present in the soil when you just put your magnet and make a play with it. So if you see in this picture, what we are going to show is, we are going to show you the filtration, and in this filtration especially we are going to separate a liquid from the solid. So if you see this entire setup here, you can see we are keeping a funnel, and this funnel is fixed to a stand, and inside this funnel there will be a filter which is nothing but a filter paper as well as a glass sieve will be there. And when you are pouring the mixture which is present in this beaker through this funnel with a filter, so the solids will get trapped here. You can see these blue particles, nothing but a solid material present inside the mixture, and the liquid is going to collected in this bottom beaker. This is a simple technique which we call as for separation. And similarly, the other instrument which we are seeing here, this is nothing but a distillation apparatus. So in this distillation apparatus, what will happen? So we are going to separate the liquid as well as what you call as soluble substances present in a mixture especially. So whenever you are preparing some aqueous extracts, for example, you are doing some research with Ayurveda and herbal formulation, in those situations what will we do? So we want to prepare the concentrated what is extracts. For this purpose, you collect some leaf material and ground them and put them in a suitable solvent and boil those material in a boiling what is called as a round bottom flask. You can see here in this picture the round bottom flask. So you are going to boil this complete what is called as mixture. So upon boiling, what will happen, the soluble solvents along with the substances, they will flow through this condenser jacket, and this condenser jacket is having a supporting system through the external water, and this external water supply which is going to cool the what is called as vapors going through this condenser, and by which the substance present inside the solvent will get collected into this beaker. Here you can see this is nothing but a pure liquid. Initially here it is having liquid with a solid dissolved in it. So like this distillation apparatus will help you to separate the soluble substances present in a mixture. So actually why we are discussing all these separation techniques in this session? So the reason being, as I have said in the beginning, so we are going to deal with the biochemistry. In the view of biochemistry, we are discussing the separation techniques. So if you see a normal typical cell which is composed of the outer membrane, internal subcellular organelles like mitochondria, Golgi complex, endoplasmic reticulum and other important vital organelles, subcellular organelles are present inside this cell. If one person is particularly interested to do a research with mitochondria, then he need to avoid all the unwanted subcellular organelles, and he want to isolate only that particular mitochondria. So for this purpose, we are going to separate the organelles. Yeah, if you see this picture here, so in this picture we can see a cell starting from a microbial cell as well as a plant cell and a animal cell. If you see all these three cells, they are having various subcellular components here, like say nucleus and DNA, as well as what is called as your endoplasmic reticulum, mitochondria. Okay, here you can see this typical animal cell. So all these cells are composed of many vital organelles. Yeah. So if you see this slide here, this slide clearly says what is the use and why you are going to do these separation techniques, what is the role of these separation techniques. So as I said, the separation techniques will give you a chance and to do the more specific, target specific research, and as well as these separation techniques will help you in performing the diagnosis and as well as drug development techniques and as well as fermentation products. If you see all these beverages which we are using these days, they are nothing but a kind of fermentation product. Even the buttermilk also, the Yakult, other products which we have purchased in the markets, they are nothing but a fermentation products. So during this fermentation, what we are going to do? So we are going to culture these microbes with other nutrients, and we are going to remove the other unwanted substances from this material, and we are going to collect the supernatant. Sometimes we will take supernatant, sometimes if required we need the protein of the same thing. But to separate these solids from the liquids, we need a specific separation technique. Another thing, for performing the forensic analysis also, sometimes we will be requiring to identify the particular type of DNA present in any blood strain, especially in the WBCs. So then also you need a precise technique which can give you the pure form of DNA from the forensic material you have identified or we have procured from the site of issue. The same time, in the coming to the food and agriculture also. In food and agriculture also, these separation techniques plays a vital role, especially the chromatographic techniques. They are going to play a significant role in identifying the adulteration in the food products. And similarly, the veterinary and poultry research also. So especially if you see nowadays, we know that milk is getting adulterated or something is getting adulterated or the dairy products are getting adulterated. So how to identify all these adulterations? So we are going to isolate the particular substance and the particular molecule present in the what is called the dairy product. The first significant technique which we need to start any isolation procedure or any basic research in biochemistry is the centrifugation. So here centrifugation is such a device where you are going to separate the solids from liquids. In simple terms, it's nothing but separation of solids from liquids. If you see the details further here, the biological centrifugation is a process that uses centrifugal force. So centrifugal force is the preliminary, what is called as the driving force behind the equipment, your centrifuge. Okay, and centrifugation is the process and centrifuge is your equipment. Here in this diagram you can see a tabletop model of centrifuge which we routinely use in our clinical laboratories and as well as the diagnosis purpose also. If you see the broad objective and the broad application of this centrifuge is nothing but to isolate, analyze the mixture of cells and as well as subcellular fractions, molecular complexes. So here are the word meaning subcellular complexes and as well as subcellular fractions. So as I said, if you just imagine the typical living cell, it is composed of many components like membrane, mitochondria, Golgi complex, ER and other things. So if you want to isolate one particular component and you want to remove the other things, then you are going to use the centrifuge. So in this centrifuge, based on the force field and based on the what is called as time and based on the type of material you are going to use, you are able to separate the different zones of the cell extract. And similarly, this centrifuge is also useful for the isolation of what is called as this antigen antibodies and protein complexes. If someone is working with RNA or DNA, so they want to isolate the pure form of DNA and pure form of RNA. So for these isolations, they will be depending on the ultracentrifugation techniques. So where they can get the distinct zones of DNA and the distinct zones of RNA to separate these biomolecules. So coming to the history of this centrifuge technique. So in the year 1920, Swedberg is the scientist who developed the technique centrifugation. You can see his picture also in this slide. So he is Mr. Swedberg who developed the technique centrifugation in the year 1920. Later on in the year 1940s, Claude further developed this technique. As I said, centrifugation, the driving force is centrifugal force, and we call the centrifugal force as relative centrifugal field, nothing but RCF. So this is such a force which is the ratio of centrifugal acceleration at a specified radius and the speed to the standard acceleration of gravity. And the unit which we give for this RCF is nothing but the RPM, revolutions per minute. So how much revolutions, how much speed it can rotate. So in this slide here you can see the basic driving force, as we said, is nothing but the centrifugal force, which is again is having a composition of both gravity and as well as the generation of the centrifugal force. The centrifugal field, which is abbreviated as G, it is always works towards the downwards of the system towards your gravity, and it is expressed as G equals to omega square R, where omega is nothing but the angular velocity and this R stands for the radial distance. So as we are discussing about this centrifugation as a significant technique, especially for the separation of these subcellular organelles. So there are some factors which will influence the centrifugation process. Like for example, denseness or the size of the biomolecule. If the molecule is big in size, it will automatically settle down very beginning. So because of this higher density, because of its higher mass, the molecule will get settles fast when compared to the others. And similarly, if the field is more, that means the force field is more, automatically it will influence the sedimentation rate of the molecules also. At the same time, if you are using a material which is highly dense, it will get settled down. Similarly, the greater the centrifugal force is, faster the particle sediments. The other thing is here, the sedimentation rate of a given particle will be 0 when the density of the particle and the surrounding medium are equal. See, here what we are trying to convey is, when you are going to use such a solvent system to separate, to perform this centrifugation, the solvent system or the medium which you are using to separate the molecules or the material, if it is having the equal density with that of your substance of target, so then their gravity will be almost 0. Here in this picture, we can see different types of rotors which we are going to use. The first one is, the leftmost one is the fixed angle rotors. Here you can see this is nothing but the fixed angle rotors. So these rotors are made up of like metal alloys, especially the spaces here you can see, these are the spaces where we are going to keep our tubes, centrifugation tubes. So why you are calling it as fixed angle means here while rotating the centrifuge, when it is centrifugation revolution, when it is rotating, the movement is fixed, the angle is fixed. At the same time, the rightmost you can see on the top, that is vertical tubing. Vertical tubing is here the difference between, if you observe the between the fixed angle and as well as the vertical tubing, you can see the tube position is here almost straight, looks like a vertical, whereas in this case it is slightly what is called as angular, which is making an angle of 14 degrees to 40 degrees angle. Here it is a straight angle. And the other model which we widely use in this clinical practices and as well as research purposes is nothing but the swinging bucket model, which you can see on the bottom side of the slide towards your left. So here you can see this is called swinging bucket model. Here I would like to explain few small important thing. In this swinging bucket model, the tube is in what is called as normal position when the centrifuge is stable. The moment centrifuge starts rotating, the tube will start lifting like this. While rotating, the tube position will be like, for example, if you imagine this is the normal position. The moment centrifuge start making rotations, this tube will take a position like this and it will make rotations. It will keep rotating. So by these rotations, what will happen? The substance present inside this test tube will be forced to move towards the bottom of the tube. The right picture, if you see the picture, this will show the differences between the what is called as angular, fixed angle and as well as your swinging bucket model. So in this first case, if you see, as we said, that is angular ones. So in this, the substance present in the tube is trying to move downwards in a angular position. If you see the movement, as you are increasing the centrifugal field, force is increasing, the substance is drastically, it is step by step it is coming down like this. Similarly, in this fixed angle, if you see the layer here, this dense layer is nothing but the substance and the bottom is nothing but a solvent, which we have as a medium. So the moment you are increasing the centrifugal field and time both, so what will happening, the substance present over the test tube, that is the top layer of the test tube, is getting sedimented. And how it is going to sediment here, you can see along the walls like this, sometimes like this, sometimes in the layers. Okay, so like this, in the case of what is called a fixed angle, the separation is in these patterns. Whereas in the case of swinging bucket model, so because of that swinging bucket position and the force which we are applying, the substance, the mixture especially, is getting separated into multiple layers, which you can see here. So why we are showing in the beginning it is in the what is called as vertical position, or the end is also vertical position, but in between it is almost like horizontal. So why? Because the swinging bucket model, we say that the moment we start rotating, the tube will take a horizontal position and it will be rotated. So far we have discussed about what is the basic principle behind the centrifugation and what are the types of rotors which we are going to use in this centrifugation. Now we will discuss something about various types of centrifugation techniques, which we call as, so here the technique is mainly depending on the apparatus. That's why we call it as different types of centrifuges. Okay, so here if you see large capacity and low speed, that's we call it as preparative centrifuge. So preparative, for example, as I said in the beginning, so we are going to isolate some subcellular components of a cell, and you want to just isolate a mitochondria and you want to isolate endoplasmic reticulum. Then you are going to take the homogenate and put on the test tube, that is centrifugal tube, and make a spin. So by this you are going to have different layers of subcellular organelles. So this is what we call as preparative. Just we are going to separate the homogenate into the distinct layers. The other one is refrigerated high-speed preparative centrifuge. The name itself says it is refrigerated. So it is having a cooling setup. Why? Because whenever the centrifugation is in the rotation, there is a chances of generation of high heat. So the high heat may lead to what is called as damage or denaturation of the substances like proteins, especially enzymes may lose their activity. So to keep their activity and their what is called as structure intact, we need to maintain the what is called the surroundings should be cool. That's why we are going to use refrigerated and cooling centrifuges. And the other one is analytical centrifuge. So analytical, the name itself says it is responsible, it is useful for the analysis. So that means you can separate as well as you can analyze. So generally we were saying about separation, but separation parallelly can be done along with the analysis also. That is the advantage of this analytical ultra centrifuge. The other one is preparative ultra centrifuge. So like this, there are majorly different types of centrifuges. We will discuss in detail one by one. Here in this picture, you can see two types of centrifuges. So the left one, the topmost one, is nothing but the tabletop centrifuge. So this tabletop centrifuge, which we generally use for the clinical purposes, where we are going to separate the serum or plasma from the blood. So in this serum plasma separation, what we do, we just collect the blood sample, and if you want to take the plasma, so you are going to run the blood sample as such, where you are going to get separation of the cells and as well as the liquid portion of the blood. That is nothing but the plasma. If you are going to add any what is called as after clotting, you will be getting the serum. Without clotting, you will be getting the plasma material. See, the difference between especially this tabletop one and as well as the high speed one is the speed. This can make an RPM of 25,000 RPM, whereas this can make an RPM of 3000 RPM. Okay, and here we can maintain a temperature of 0 to 40 degrees. Okay, whereas in a normal one we cannot maintain the regulate the temperature, whereas in the cooling one we can maintain the temperature also. And especially this high speed, the moment we say the word high speed will enable you to separate individual subcellular components, whereas the tabletop one will enable you to separate just total cells but not the individual subcellular components. But whereas high speed one will enable you to separate individual subcellular components like mitochondria, Golgi complex, ER and other things. Yeah, so moving further, this is ultra centrifuge. So ultra means it is having much higher speeds. Okay, here you can see the RPM of this one is 75,000 RPM. Okay, and here we can separate like you can proteins and other carbohydrates, glycogen and other polysaccharides you can separate by using this ultra centrifuge. So here in this picture we are trying to distinguish the types of preparative centrifugation. So one is, you can see the top one is nothing but a homogeneous medium with differential centrifugation. The other one is, the bottom one is density gradient centrifugation. So here both are preparative centrifugations, but the difference between the top one and the bottom one is, the top one, if you see here in the beginning, the very beginning, the mixture means the molecules, you can say the substances, they are mixed together, and the moment you start performing centrifugation, the moment you have increased the time and as well as the centrifugal field, you can see here the mixture, the substances present in this mixture, they are going to be grouped. Okay, like small sized particles will be top layer and the medium size will be in the middle layer and the large particles will be in the bottom layer. But here we are not able to make clear cut distinguishing zones. But however, the reason is here the medium which we are going to use, this is homogeneous medium. So because of the homogeneous medium, the substance, the components present in the mixture, they are separating based on purely their sizes. Because that's why smaller ones are in the top and the larger ones are in the bottom of the test tube. But whereas as you see the bottom one, that is a density gradient centrifugation method, here what is happening, the medium which we are using, which is having a density gradient, that's why the substances are getting separated into individual components. You can see the distinguished clear cut zones are there, smaller, medium and higher. So further, if you go to this density gradient centrifugation, this density gradient centrifugation can be further divided into two types. So one is rate zonal, the other one is isopycnic. So as we said, rate zonal means here we can have different sizes of what is called as densities of the medium. So based on the density, the substances will get stacked up, separated. In isopycnic, the similar molecules, similar density molecules will get stacked up in this isopycnic case. And moving to the analytical ultra centrifugation. So in this analytical ultra centrifugation, so what is happening, if you see this picture clearly here, This is nothing but the rotor. In this rotor, we are having two what is called as spaces. One space is for the test sample, the other space is for the reference sample. That means when you are going to make centrifugation, separation, so the light will pass through both the centrifugation tubes, but the major advantage of this analytical centrifugation is separation along with analysis. That means while separating itself, you can say whether this material is consisting of a particular desired substance or not. And this reference space where you are going to keep the reference tube, and this will give you a reference value where you can compare unknown sample with the reference sample. And if you see, the determination of purity of macromolecules is one of the advantages. The other one is the determination of the relative molecular masses of the solutes in their native state. That means while separating itself, you can make a what is called as analysis also. And the examination of changes in the molecular mass of supramolecular complexes, and the detection of conformational changes, and as well as ligand binding studies. So here these ligand binding studies are nothing but, so for example, you are developing some drug against a particular protein, a particular disease. So generally many of these drugs are nothing but ligands. Okay, so and these drugs will interact with particular proteins. So for this purpose, we are going to use this analytical ultracentrifugation. So here by this we are going to conclude the session of separation techniques 1, and these are the few references which you can refer, and in the next session we will be talking about separation techniques 2, which is based on the chromatography. Thank you one and all.